Hey everybody, I'm Gregory Johnson. I'm one of Bob's PhD students, and I will be discussing basically how the non-parametric diffeomorphic model works in Cell Organizer and why we find it very useful. What Bob went over, what we call a diffeomorphic model uses a metric called large deformation diffeomorphic metric mapping. And this has a bunch of properties associated with it, which make it really, really good for our application of modeling cells and the joint distribution of compartments within those cells in a way that allows you to sample new instances that are topologically constrained, as you will see. And with by measuring the distances between multiple shapes, we can then project the distance matrix into what we call as a shape space, which is a reconstruction of the underlying coordinates from which the data arose. And then we can treat those coordinates like any feature space or like a Euclidean space. So when people build models of dynamics, like people running, and this is performed in some Fourier space, where you can see cyclic behavior, we can also put these cells in a similar space and model how they move. But we get the advantage of this by that we can synthesize instances according to that model, rather than instead if you were using morphological features, you wouldn't necessarily be able to synthesize instances of cells. So we have lots of models and in Cell Organizer, and there's been lots of models in literature. Why would we be interested in using a what is maybe somewhat complex and computationally expensive method? So first of all, cells don't always satisfy the assumptions of parametric models. So Bob briefly talked about this ratio model of how the distance of the cell boundary from the nuclear boundary. And if we have a segmented neuronal cell, that may not be well represented with this parametric model. For example, this doesn't accurately model neurites. And we see that the nucleus here is of somewhat complex topology. And the model, although it gets the general shape, it doesn't get that in very high detail. In literature in the last few years, there's also been these parametric shape space models. So we can see here some images of fish scale cells that people use to study motility. And you can draw a polygon around each of these images of a cell to get a model of the cell shape. And then you can project these polygons into a lower dimensional space and study the modes of variation across that space. So here is one such mode where we see that essentially the cell is increasing in size. And this is exactly what we would like to do as well. And additionally, you can look at another mode of variation, and you see that it sort of turns from a circular shape to an elongated shape. But there are limitations for this outline model given particularly complex shape. So if you have a shape of a turtle and you want to turn it into a starfish, the correspondences from locations on the polygon as you interpolate from one polygon to another, may not give you something that's topologically sound. So it may form disjoint regions, or it may not form a shape that is representative of any cell. And so, of course, we see cells with a very complex topology. And I performed an experiment with using this model on images of PC12 segmentations and sampled from them. So I just modeled this distribution as a Gaussian mixture model and sampled. And here I have 5 example images ordered by the distance from the center of this distribution. And you can see that the center of the distribution models cell shapes pretty accurately. But as we get to the lower density areas, we get cell shapes where the nucleus is not necessarily inside of the cell, or there are regions that are sort of broken apart from the rest of the cell shape. And we would like to have a model that allows us to continuously move from one cell shape to another, but preserve appropriate cell shape topology. So this begins our journey with LDDMM. This is a method that was originally used to study spaces of shapes with MRI segmentations, such as looking at how the shapes of different brains are related. And so first of all, what is a diffeomorphism? So this is a smooth and invertible mapping from one coordinate space to another. Here's an example of a diffeomorphic mapping from a regular rectangular grid to a distorted surface. And you can think of this as something like taking a map, a two-dimensional map, and mapping it so features correspond in a one-to-one manner from a two-dimensional shape to maybe a projection, a 2D projection of a globe. So this mapping preserves the shape, or this mapping allows you to correspond one feature to another feature in 2 sets of images. An example of which would be this. So we have 2 images, one of a neutral face and another image of a smiley face, and we want to find a smooth transition from one of these images to another. And this, like what Bob said earlier, this method was later developed to study nuclear morphometry. And we can't just interpolate these images as if they were vectors. So if we have an image of 2 faces, and we smoothly transition from one image to another, you may get an intermediate that is somewhat nonsensical in your context. But if instead you can map features to each other, such as, in this example, eyes and nose, then you can interpolate between these 2 images and get I guess, a morphologically valid interpolant. And this example we already saw with Bob, where we essentially push and pull at different locations of these images such that we can smoothly transition from one shape to another. And this pushing and pulling is according to a minimal energy transformation with respect to the gradient of the deformation field. So if we look at these two images again, you may think that this point here is being moved in a straight line to where the corresponding feature is in the next image. But that is not, in fact, the case. This transformation is stepwise and nonlinear from time point to time point. So such that this point moves in sort of an arc that minimizes the total, and the rest of these points move in arcs that minimize the total energy spent to deform one shape to another. And then in our particular application, we can use this for measuring the distances between cell shapes or interpolating from one cell shape to another. So here's an example of two HeLa cells. And you can see that, or if I have this image here and the other image at the end, even though we don't have any intermediate shapes, we can synthesize images that are topologically sound. And as a person who looks at images of cells all day, every day, I would not be able to tell you that any of these intermediate shapes are artificial in any way. A further advantage of this is when we measure a multitude of distances and form a complete distance matrix, after embedding, this space models the joint distribution between cells across morphological features. So what that means is that we don't need to come up with models for how the nucleus varies and the cell varies, because this model contains how that information varies and how that information co-varies as you move from cell to cell. So in this particular example, we see cells on this side that are perhaps rounder. And as you move over here, they get smaller. And down here, maybe the cells are more square, and up here, they're more triangular. And you can see that the nucleus varies in a smooth manner across this shape space as well, along with the cell shape. So now I'm going to get into how this diffeomorphic model is constructed. So to reiterate what Bob said earlier, we measure the distances from one cell to another and start populating a distance matrix. We then use multi-dimensional scaling to embed this in a reconstruction of the original space from which the cells were drawn. And this, of course, is an approximation of the original space, because we don't know what that is to begin with. But this is a somewhat expensive computational task. And measuring the distances between two shapes, such as, especially if they're high-resolution images, which these are, may take a long time. So it could be from a couple of minutes to a couple of hours. And if you want to measure the distances between 1,000 cells, that's basically 1,000 squared times the amount of time it's going to take for your computation. So that may be hundreds of thousands of minutes or hours to complete this matrix. So what we have currently implemented in Cell Organizer is to go down the columns of the matrix to give you a complete shape space to allow the user to get the most complete shape space for whatever time you've committed to computation. So here we compute the distance between this orange cell and this turquoise cell. And then we compute the distance between the orange cell and this purple cell, and then the turquoise cell and this purple cell. And although we are training on a subset of the data, this gives us a complete and valid distance matrix so this would be the valid region of this distance matrix. And then we can use the same methodology as before to embed the best model available into the shape space. This shape space, of course, becomes more descriptive the more data that you train it on. So there are also alternate methods, such as landmark multidimensional scaling, where in the method that Bob described, we measure the distance from one cell to all other cells. And this continues. So now we have, so this is treated as a landmark cell. The next cell is another landmark cell. And this goes on. But of course, filling out a very large matrix will take a lot of time. So we want to be able to approximate a region of this distance matrix, even though we may not have seen it. And this is relatively simple with what is called the Nystrom approximation. And this, of course, gives us an approximation to this area, to this region, because when we measure the actual distances, there may be noise, or it may be slightly non-Euclidean. But this, again, allows us to, instead of using a subset of the data, would allow us to build the shape space with the complete set of cells that we have. So once we have a shape space, how do we synthesize new instances? So you could imagine that we have 4 cells embedded in a 3-dimensional space. And this defines a simplex. And anywhere within the convex hull of this simplex, so basically inside this pyramid, we can synthesize a novel or an in silico cell. And so let's say we want to synthesize a cell at this point. Because this diffeomorphic distance is only computable between pairs of cells, we iteratively synthesize artificial cells along the edges of this simplex. So we first compute this cell, and then we can compute an intermediate cell between this synthetic cell and this observed cell that obeys this diffeomorphic distance. And then using this synthetic cell and this observed cell, we can then synthesize another point with an in silico, a novel in silico cell inside a simplex of an arbitrary dimensionality. And of course, we've been talking a lot about statistical models and probability distributions. This is an example of a shape space of about 100 HeLa cells. And I would like to know the probability of there being a cell, the probability density of a cell at this location is indicated by the X. So using a triangulation method called Delaunay triangulation, we can form simplices inside this 2-dimensional shape space. But of course, this method works equally well in spaces of arbitrary dimensionality. And you can see here this triangulation forms a convex hull within which we can sample any cell shape. And to model the probability density here, it is intuitive to think that each simplex has equal probability mass. And so therefore, the probability density is 1 over the volume of the simplex in which your new synthesized data point is in, times the number, 1 over the volume times the number of simplices. And this is a entirely non-parametric probability distribution. And the advantage of this is that you don't assume a prior over the what you'd expect the shape to look like. And this probability distribution increases in accuracy as you acquire more examples. And so, of course, we can use the same methodology as before to synthesize an instance within the space. And because we can treat this space like any feature vector or any feature vector or a Euclidean space, we can also parameterize it in a parametric way. And I'm going to show you some demos of doing this later today. But I've modeled the space with between 1 and 4 Gaussian distributions using expectation maximization of a Gaussian mixture model. And we can additionally perform statistics on this fit of a Gaussian mixture model and pick one that satisfies some criteria. So you can see here that a single component Gaussian gets this big group very, very well. And a 2-component Gaussian mixture model not only models this large cluster here, but also this little group way off by themselves. And considering this, you may lead you to believe that there's these 2 modes of population within this distribution. And as we increase the number of components in the Gaussian mixture model, this distribution becomes more complicated. This distribution here turns out to be the best using AIC and BIC, which are 2 metrics used for model selection. So this is all well and good. We have this gives us a feature space which does not define features explicitly, but rather learns features that correspond to the morphology of the cell. The feature dimensions, so the first you see in the previous slide, you'll say like component 1 or dimension 1. These correspond to the dimensions with the highest eigenvalues. So that basically means the ordered dimensions that account for the most variance in this distribution. And in addition to being treated exactly like a normal feature space, we get the added advantage of synthesis capabilities anywhere in the space. And so now that you know how to compute a diffeomorphic space, we can then perform all types of analyses on the images and data that we have available. So here is a HeLa shape space with the cells colored by DNA intensity. And we can see that the first component, using linear regression, we can see that the first component has a pretty poor fit for describing how the morphology changes with respect to cell intensity. And likewise for component 2. But this third morphological component has a very, very high degree of fit with how the DNA intensity changes. So perhaps this dimension of our feature space gives us a lot of information about where the cell is in its cell cycle from just morphological information alone. Like Bob showed earlier, we can perform random walks inside this shape space using some parameters that we have had manually selected. But if you have unordered time series data, let me pause this. If you have unordered time series data, so you have a collection of cells available at different time points. So you've run an experiment and you give your cells some drugs and you wait some time. And then you look at the cells and again, and you wait some time, and you look at the cells again. You have cells at different degrees of influence under the condition. And you want to be able to construct a pathway that allows you to not only know the general trend of how this condition has an effect on the cell, but you want to know about heterogeneity in this population. So if cells with certain morphological features, do they happen to have a particular set of behaviors with respect to the condition applied? So you can construct what would be called a minimum energy pathway between sets of images of cells. So consider that this collection of points is a set of cells within our shape space. And we know that this set of cells is taken at time 1. And then another set of cells taken at time 2. Using an assumption of minimal flow, so basically a minimal distance traveled to get from one time point to another in the shape space, we can construct correspondences of cells from one time point to the next. So if you have a more complicated shape space, you can, and this is basically, if you look at your figure, your two-dimensional projection of your shape space, and you see a trend of the cells moving across like this, you can reconstruct pathways that use intermediate shapes that minimize the total net flow of your system, and then allow you to allow you to compute statistics on given a cell in a particular location in the shape space, what will you look like forward in time and backward in time, given your morphology, and then be able to evaluate how the heterogeneity of cell morphology affects the response to your condition. Additionally, this diffeomorphic metric implies a deformation field over the cell. So if we have an image of two cells and they have a different protein pattern stained in them, and we want to know how this protein pattern would look like in this other cell shape, we can propagate the protein patterns according to this deformation field to give us what may be a good representation of the protein pattern in the next image. And so here's an example, a video of one cell morphing into another cell with the protein distribution going along for the ride. And that's almost exactly my 30 minutes. Um, I'll be explaining how to do many of these methods in the tutorial later today, so I hope to see you guys there. Thank you. Yes?
So how do you go from this representation that you have to, say, if you want a 3D, can you?
A three-dimensional space or three-dimensional images. So the diffeomorphic metric, as it's currently implemented, accepts cell images that are 2D or 3D. But when you compute, that should not be confused with when you compute this distance matrix and then project it into a space of some dimensionality. So in multi-dimensional scaling, the function in MATLAB, you put in your distance matrix, and it returns another matrix of the same size. And the columns of this matrix correspond to the coordinates in an embedding of that dimensionality. So the first column would be the coordinates in the first dimension. The second column would be the embedding in the second dimension, or the third, or the fourth, or the fifth. And you can think of this as all the images live on a hyperplane of very complex geometry. And this, by measuring the distances from all the cells on this plane, we can then reconstruct that hyperplane with some embedding. And there's, of course, other methods to do this embedding. We just happen to like MDS because it's fast and returns good results. Any other questions? All right. Thank you very much.
I think we'll take a little break.